Welcome to this lecture on the fundamentals of biomolecules. Today, we are going to explore a crucial concept known as the glycosidic linkage. If you have ever wondered how plants build strong trunks, or how our bodies store energy from the food we eat, the answer lies in this tiny, invisible chemical bond. Let us dive in and discover how simple sugars connect to form the complex structures of life. To truly understand this concept, imagine you are playing with a set of Lego blocks. Each individual block represents a single, simple sugar molecule, like glucose. By itself, a single block is useful, but you cannot build a massive, complex castle without connecting them together. In the biological world, cells need to build large, complex structures to store energy or create strong cell walls. To do this, they must snap these simple sugar blocks together using a very specific type of chemical connection. This vital connection, the biological equivalent of the locking bumps on a Lego piece, is what we call a glycosidic linkage. Now that we have our Lego analogy, let us look at the formal scientific definition. A glycosidic linkage is a specific type of covalent bond that joins a carbohydrate molecule to another group. This bond is formed when the highly reactive hemiacetal or hemiketal carbon of one sugar molecule reacts with the hydroxyl group of another molecule. Because this chemical reaction releases a single molecule of water as a byproduct, it is formally classified as a dehydration synthesis or a condensation reaction. It is the fundamental bridge that allows tiny sugar molecules to join hands. Building on that definition, we must focus on the specific carbon atom that participates in this bond, which is called the anomeric carbon. The spatial position of this anomeric carbon is incredibly important because it determines whether the resulting linkage is an alpha or a beta type. Understanding this geometry is crucial. Why? Because the exact shape of this bond dictates whether the resulting sugar can be easily digested by humans or if it is completely indigestible. A tiny shift in spatial arrangement completely changes how the molecule behaves in nature. This brings us to why we care so much about this specific chemical bond in our daily lives. The glycosidic linkage is literally the glue that holds our primary energy sources together. When you eat a bowl of rice or a potato, you are consuming starch, which is just thousands of glucose molecules held together by alpha glycosidic linkages. Your digestive enzymes are specifically designed to act like molecular scissors, cutting these exact linkages to release glucose for energy. Furthermore, plants use a slightly different version, the beta glycosidic linkage, to build cellulose, which gives wood its strength. Now that we understand the real-world impact, let us look at the chemical representation of this linkage. In biochemistry, we represent this formation as a chemical equation that perfectly balances the atoms. On the left, we have two molecules of C6H12O6. Each represents a single monosaccharide, such as glucose. When they react, they do not simply add all their atoms together perfectly. Instead, the formation requires the removal of two hydrogen atoms and one oxygen atom, which combine to form the water molecule, H2O, seen on the right. The resulting disaccharide is C12H22O11, showing that it is exactly one water molecule lighter than the sum of its two parts. Before we move on, let us see this chemical equation in action with a concrete example, the formation of maltose. We are given two separate molecules of alpha-D glucose. Our goal is to understand how they combine. First, we identify the reactive groups. We look at the first carbon, the anomeric carbon of the first glucose, and the fourth carbon of the second glucose. The enzyme facilitates a reaction where the hydroxyl groups on these specific carbons interact. By removing a water molecule, the remaining oxygen atom acts as a bridge, connecting carbon 1 to carbon 4. This creates what we specifically call an alpha-1,4 glycosidic bond, resulting in maltose and water. Following the formation of maltose, we must note a very important chemical property. In the maltose molecule we just built, the glycosidic linkage only ties up the anomeric carbon of the first glucose molecule. The second glucose molecule still has its own free anomeric carbon at its far end. Because this reactive center is untouched, the sugar ring can still open up and participate in further chemical reactions. This means maltose remains a reducing sugar. It can still react with other molecules or extend the carbohydrate chain even further, which is essential for building long starch molecules. Let us visualize exactly what we just discussed by looking at this diagram. On the left, you see two individual alpha-D glucose molecules. Notice the highlighted areas, the hydroxyl group pointing downwards on carbon 1 of the first molecule, and the hydroxyl group on carbon 4 of the second molecule. The arrow in the middle represents the dehydration synthesis reaction. As we move to the right, you can see the final maltose molecule. The two sugar rings are now permanently connected by that single oxygen atom acting as a bridge. This is the alpha-1,4 glycosidic linkage. You can also see the water molecule that was released during this process. Now that we have successfully built maltose, let us look at a second scenario, the formation of ordinary table sugar, known as sucrose. This time, we are given two entirely different monosaccharides, one molecule of glucose and one molecule of fructose. We must carefully examine their anomeric carbons. For glucose, it is carbon 1, and for fructose, it is carbon 2. During the dehydration synthesis, the glycosidic linkage forms directly between these two highly reactive anomeric carbons. By removing water, we lock both of them into the bond simultaneously, creating an alpha-1 beta-2 linkage. This unique bond in sucrose leads us to a fascinating biological consequence. Unlike maltose, where one anomeric carbon remained free, the glycosidic linkage in sucrose ties up both reactive centers simultaneously. Because both anomeric carbons are locked into the bond, neither sugar ring can open up to act as a reducing agent. This is exactly why sucrose is perfectly stable and is classified as a non-reducing sugar. This chemical stability makes it an ideal unreactive molecule for plants to transport energy through their sap without accidentally reacting with other cellular components along the way. We have covered the full topic of glycosidic linkages, so let us review the key takeaways. First, remember that glycosidic linkages are the essential covalent bonds that connect simple sugars into complex carbohydrates. Second, they are formed through a dehydration synthesis reaction that removes water to create a strong oxygen bridge. Finally, the specific carbons involved and their spatial arrangement, whether alpha or beta, completely determine the physical properties of the carbohydrate. Mastering this concept allows you to understand how nature builds everything from the starch in a potato to the rigid cellulose in a tree trunk. Great job following along.